Good morning, everybody. So may I ask you please to take your seats. So we will start with the plenary session of today's. So on behalf of the Mexican Materials Society, it is my great pleasure to introduce our plenary speaker, Molly Stevens. My name is Juan Muñoz. I am professor from CIMESTAB in Querétaro. Well, let me just read very briefly Molly's bio. Molly is Dame Commander of the Order of the British Empire, Fellow of the Royal Academy of Engineering, and Fellow of the Royal Society, as well as John Black Professor of Bionanoscience at the University of Oxford. She holds part-time professorship at the Imperial College London and the Karolinska Institute. Molly's multidisciplinary research has sought to balance fundamental scientific research with technology development to address some of today's major healthcare challenges. The impact of her work influences research groups around the world, which is demonstrated in her more than 450 papers, which have been highly cited, reaching over 50,000 citations, and she has an H index of 109. Molly has been also recognized in 2018, 21, 23 by Clarivate Analytics as highly cited researcher in a cross-reference or a cross-field research. So Molly is also a serial entrepreneur and the founder of several companies in the diagnostics, advanced therapeutics, and regenerative medicine space. Her work has been instrumental in elucidating the biomaterials interfaces, and she has created a broad portfolio of designer biomaterials for applications in disease diagnostics and regenerative medicine. So today's talk of Molly is innovating materials design to transform disease diagnosis and treatment. So, dear Molly, on behalf of this community, I want to let you know that we are really excited about your talk, which I am pretty sure it's going to be very inspiring. And so the floor is yours, Molly, please.
Thank you. Thank you. Thank you so much. Could I have my slides, please? Are you able to put the slides? Let me reshare them. Let's see if that works. Okay, perfect. Thanks so much. I mean, what a location. I just couldn't believe it when I arrived here, and I thought, wow, I'm coming to this conference every year for the science and for the location and the wonderful community. It's truly, truly an honor to be here, and a perfect end for the summer. So I'm going to be talking about some of our biomaterials platforms that have a number of different application areas. And I'll be today just giving you a little flavor of some of the things that we've been doing within our multidisciplinary group. You can see the group comes from all around the world, including from Mexico, actually, although I would say more Mexicans would be great, because we don't get so many applications from Central and South America, and we're very keen to have an incredibly diverse group, both in terms of nationalities, but also backgrounds. So we have people with engineering backgrounds, and chemists, and cell biologists, and physicists, and surgeons. And it's this very, very interdisciplinary research that we do. And we go from fundamental science right through to applications. So I'll be talking today as well about some of the application areas. And you can see some of our spin-out companies there in the bottom right that are in the area of diagnostics and also advanced therapeutics. And I'll mention a couple of those, just a couple of those today. So we moved in April 2024. After 20 years at Imperial College, we moved to Oxford University. I'm helping to head up this new Kavli Institute there, and we have the whole top floor of this new building, and it's really, I think, really transformative for our research environment, and I'm really, really looking forward to our new adventures there. The entire group moved with me from Imperial, so there's some familiarity. We have the same group, but completely new surroundings, new collaborators, new possibilities, and it's very exciting. So we work at interfaces, so interfaces between materials and biology, and you can apply that in different areas, for example, in tissue repair and advanced therapeutics, but also in early disease diagnosis. And then underpinning all this, we're very, very interested in characterization. So thinking about new methods that can help us to characterize biomaterials and tissues and the way that cells interact with materials. And we've even invented some of our own new techniques, and I'll mention a couple of those today. So if we look at a field like regenerative medicine, all the organs you can think of, the musculoskeletal system, the brain and cardiac and so on, there are lots and lots of clinical needs for this, and actually groups around the world working on all these different tissues and thinking about different material strategies to help. And it's a complicated area. And so Our group itself is, of course, very collaborative, but we also work much wider than within our group. And a few years ago, we set up this UK regenerative medicine program, Smart Materials Hub. And I've been serving as the director of this, and it brings together 11 different universities across the UK. And the reason it does that is we need a critical mass to address the really difficult challenges in translating biomaterials from the lab bench through to clinic. So this hub works on different types of biomaterials. It works on printed hydrogel systems, templated porous structures, gradient materials. It works on nanopattern materials, all sorts of different materials. But what it does that's very different to a normal academic center is that we also set up, right in the beginning of this hub, we set up committees and advisory groups that would help us with translation. So groups that would help advise us on safety and immunology and manufacturing and scale-up and regulatory issues. And this means that all the researchers coming into this hub, right from day one, are looking at the target product profiles for the materials that they're going to be inventing and translating. And this has been really effective. It's led to many different spin-outs from the hub and also 3 different human clinical trials being planned at the moment. So this kind of critical mass around thinking around translation is important. And this is because it's really difficult as well to get the right design of your materials if they're going to translate. It's very, very easy to make a material that you publish in an academic paper. It's much, much more difficult to make a material that's going to end up in clinical use and helping a lot of people. And so one of the things we think about all the time in our group, and these are just some opinion articles that we wrote recently, is thinking about the right level of complexity that you put into your biomaterials design. Because if you make the material too complicated, it might be difficult to translate. And then if you don't give it enough biological function, it may not work well enough. And so that balance is a very, very difficult thing to get right. And so we also are constantly turning to more fundamental studies to understand what is the level of complexity that we require. So this is one of our very fundamental studies where we look at the nanoscale organization of bone tissue. Bone, of course, is a biomaterial. And we zoom right in with electron microscopy. This is in collaboration with Roland Kroger at York. And we attempt to understand better the nanoscale organization between the organic and the inorganic portions of bone. And we were able to uncover design principles that had not previously been reported, and also to redefine the levels of hierarchy within bone up to 12 orders of magnitude, which is more than had ever previously been reported for bone. But this very fundamental science paper actually led to a lot more. It led us to understanding that nanoscale structure and then designing engineered materials that had more nanoscale organization in the organic and inorganic components. And so fundamental insight then leads us to biomaterials design that we can use for bone tissue engineering. Similarly, in this study here, this started off very, very fundamental. So it was a study looking at patterning of single cells. Here's a stem cell patterned as a triangle. And these were patterned because we knew that if you patterned a stem cell as a triangle, it's more likely to become a bone cell when it specializes. If you pattern it instead as a circle with the same surface area, it's more likely to differentiate down in what's called an adipogenic route, so towards a fat cell specialization. But we didn't understand why, and nobody in the field really understood why. And so our contribution to this field, where many people are working, was to look at these cells, to pattern them, and then to try and understand the interface between the cell and the material underneath. And we did that using focused ion beam-based imaging and then reconstruction of the underneath membrane of the cell. And so what we're looking at in these images here at the top is the underneath membrane of a triangle cell, and at the bottom of a cell patterned as a circle with exactly the same surface area. And you can see those membranes, even though they have the same contact surface area, are really, really different. So the one that's patterned as a triangle has all these invaginations. That leads to very, very important biophysical changes that lead to a lot of cascades of biological information that then lead to that particular form of differentiation. And so again, very fundamental study, but what can you learn from this? Well, actually, you can then take these kind of patterns and put them, for example, onto orthopedic implants and things like that to help to control the way that cells might differentiate. We also work on nanoparticles, essentially, for different types of drug delivery. And there are many, many different types of nanoparticles that you can use. So I know not everyone in this audience is in the biomaterials field, but I saw quite a few studies actually in the posters last night that were involving some of these types of particle systems. And you can essentially make them from lipids, so lipid nanoparticles, or from polymers, or use other native biologically derived vesicles. And we work on many of these different types of particle systems. And I'm going to tell you in a second some of the things we've been doing around better understanding the characterization of these particle systems. But before I do that, just want to give you an example or two of our very recent work on particles so you can get an idea of the sort of things we've been looking at. So I just picked a couple of the most recent papers that we've published. The top one is from three wonderful postdocs who are now all professors, so Haimin, Jono, and Adrian. And what they did was to develop polymer-based particles that could help control the rate at which heart cells could beat. So this is an optogenetic study. And in the bottom example, what Adrian there has done is to develop polymer-lipid nanomimic particles to stop viruses and also malaria parasites from entering into red blood cells. And so it's around particle design for different types of biological application. We also do slightly more fundamental things. And so some of the work we've been doing has been inspired by biology. A lot of what we do actually is inspired by biology. So thinking about something as exciting as a circadian rhythm, so how we all respond to cycles of light and dark. And we posed ourselves the question of whether we could artificially mimic the sorts of things we have in our body that help in circadian rhythm processing. So for example, could we mimic the photoreceptor-coupled transmembrane channels of living cells? And so this was actually a very, very fundamental study. This was led by Omar, just published just over a year ago. And Omar was a postdoc at the group. He's just recently started as an independent professor now in London. And what Omar was interested in doing was developing different populations of polymer-based nanoparticles that could communicate in really interesting ways with each other. And in particular, they could provide negative feedback loops in red there and positive feedback loops. Now, both of these types of particles contain antagonistic enzymes. So the ones on the left there contain an enzyme called urease, and on the right, an enzyme called esterase. And between them, they can chemically communicate. And the ones on the right are controlled by light. The permeability is controlled by light. And we can get this photo-switchable gating of non-equilibrium enzymatic feedback. And what you end up being able to do with these systems is actually to control the pH of the environment. So it starts off very fundamental, thinking about artificial organelles, maybe soft robotics. And then the fact that you can control the pH through light, through the activity of these enzymes, means that you can actually do these to do chemomechanical work. And so what we did is we put these particles in the presence of gel systems that could swell and deswell in different pHs. And now those systems can get activated by light through the communication through our polymerasome-based systems. We also can think about some pretty translational work in the form of this spin-out that we recently founded, which is a spin-out called NanoGrab. And what we're doing there is actually combining our experimental work with computational drug discovery to essentially create binders using AI, but binder systems that can be multivalent and can be specific, but can also, you can actually control very well the degree to which they interact with the target. And the idea here is these can function as drugs by themselves, but we can also use them to decorate the types of nanoparticle systems that I've been showing you so they can do better targeting within the body. Okay, so just showing you a few examples of some particle systems that we work on. Now, for all of these particle systems, a challenge that we've had, and actually a challenge the entire field has had, is the ability to actually study these, like experimentally study these and understand the heterogeneity and understand what's happening at the nanoscale within the individual particle systems. So a few years ago, I set the challenge to my group of, let's develop a technique that can enable us to trap these individual nanoparticles and measure their chemistry in an automated way. And that automation of that sort of approach had never been previously done. And so we developed a technology that's now spun out into this company, SPARTA Biodiscovery, called single particle automated Raman trapping analysis. And what this does is we can trap our nanoparticles from a bulk population of nanoparticles with a laser. We can measure their chemistry with Raman spectroscopy and understand the composition and also the heterogeneity of our nanoparticle systems. And we can look at very complicated mixtures, but study the nanoparticles down at single nanoparticle resolution, all in this automated way. So you can get information about hundreds, if not thousands of particles within your sample. And you can also correlate this to size. And when we published this back in 2018, this was essentially what's called a breadboard setup. So it was in a dark room. So one of our labs that was, had a dark room. You could only go in if you had the laser safety training. You had to be essentially one of the physicists within our group. And it was on this optical tables, a big breadboard setup. And we published this in Nature Com, and it was essentially technology that could trap the particles, measure their chemistry, correlate that to their size by displacement of a Raman active solution marker, and even do online dynamic reaction monitoring, all powered by our own code, our own software. So this is where the computer scientists in the group come in really useful. But it was only able to be used by specialist physicists within the group. So in the few years following that, we've worked really, really hard to make this accessible to everybody. So people with more biological backgrounds, people in open labs that don't have access to dark rooms and so on, and have made this now into this benchtop instrument that you just saw in the animated video. And it's really the first instrument in the world that can do that rapid single particle analysis at the touch of a button. So it looks like this. You can fit it just on a normal benchtop. It's now just, as of this year, received the CE mark approval across Europe, which we're really excited about. So this is now in commercial sales and has many, many different pharma companies utilizing this technology, as well as some academic groups. So these are some of the things we've been looking at. On the left, you can see the different classes of particles. I hope this is useful to many people in the audience here. We can also, of course, do non-biological applications. I'm just showing here some of the biomedical applications, because that's what my group has been focusing on. But you can use this as a generic chemical analytical technique. We can look at the functionalization of our particles, how much drug is loaded, if there's any contamination, and so on. And we've done lots and lots of work on this, but I'm just going to show you a couple of examples. So I want to show you this example here, because this was an experiment that I was so excited about, because it was the first time that we trapped a particle and then measured real-time changes in its chemistry. And what we're doing here is if you look at the top left, we're trapping a polystyrene particle we're introducing an alkyne moiety, and that leads, to a peak in the Raman spectra that's very easy to recognize. And then we do a secondary reaction to convert this alkyne to a triazole. And what you see there is very exciting because you see that alkyne peak disappearing, and then you see your essentially signature related to the triazole appearing. So you have this real-time monitoring of a change in chemistry within a single particle. And I'd be very, very excited to try this for lots of different other chemical reactions too. And if people have ideas of how they could use this within their own systems, I'm very excited to hear about that. We can also use it to study lipid nanoparticles. So this was a study led by Hannah, a postdoc in our group who's now also just started her own independent group. And what Hannah was interested in was looking at different types of lipid nanoparticles. So these are similar to the kind of lipid nanoparticles that were used for a couple of the very successful COVID vaccines. And what she could do with the SPARTA is to trap them and to measure compositional differences, but also to measure how they responded to enzymes like phospholipase D that are present in the body in different disease states. And what the phospholipase D can do is to cleave off the head group, and then you can monitor that dynamic reaction in real time as you convert DOPC to DOPA. So really, really interesting in terms of also understanding some of the mechanism of how these types of particles might behave in the body. And you can do other things too, like understand how proteins bind to them and all sorts of interesting mechanistic questions. So we also have great involvement in this company called NanoVation Therapeutics, which was actually set up by an incredible academic called Peter Collis, and there's 5 other academics, including myself, involved in this. And the main base is in Canada, and then we have a wholly owned UK subsidiary in Oxford, as well as a base in Switzerland. And this is really around lots of design of different lipid nanoparticle formulations, particularly for gene therapies. And this company now is grown extremely rapidly. This is a very hot area of research. Okay, so I've shown you the kind of drug delivery systems that have been studied by many people for a long time. And what I want to show you now is some new particle systems that we've been developing in our lab using new bioinks that we've invented within the group, and also very high resolution printing. So this is a particle that looks spherical, but actually if you zoom in on it, it's got an exquisite control of the morphology and topography of the particle. These are made from polymers. And we've not only made these sort of particles, but we've also printed systems that can fill the particles very accurately with different types of drug cargo. And so what you can see here in orange in the top schematic is our particles, and then in blue is a system that we've printed that can enable us to do a microfluidic loading of those particles. And then we have a dip sealing process that means you can fill them with exactly the right amount of cargo. So this video will show you here how we fill these little particles. And then on the side here, you can see actually a cross-section on the right there of the particle, and then you'll see these guys filling up. So you have complete control over the amount of cargo that you put in there. And that was published, as you saw, in Advanced Materials quite recently. But what we then wanted to do was to think about how you could have those kind of systems, but maybe move them in interesting ways, and maybe have them release drugs on demand in interesting ways. And I asked my student to think about some really bio-inspired mechanisms for how you might release drugs from them. And he came back with this video here of the puffball fungus, where if this fungus is contacted by even a drop of water, it then releases all of its spores. And so we developed new types of particles that had on-demand drug delivery, with essentially a cap layer, which is also made from polymer, but it is responsive to near-IR light and lasers, and it will essentially melt when you apply near-IR light to it and release drugs on demand. So the particle itself, you can see in the bottom right, it's just some different design systems, and then we have a selective sealable layer that we can activate so it melts on demand and lets drugs out. And if you do that, you can then get kind of pulsatile release of drugs on demand. So you can see in the top right there, we have the system that can be activated by light, and you will start to release drug, and you can do this for multiple different cycles. And this was work led by a different student in the group, so this was led by Shin, who's actually a very incredible student. I mean, he finished his PhD and got a professorship straight away, so without even a postdoc, which, as I'm sure you're aware, doesn't happen very often. This is a very, very remarkable student. And once Shin had finished this work, he came into our group meeting and he said, actually, I want to also get these things to kind of move around in interesting ways too. And so he introduced some magnetic components to them and made sort of double particle layers. This is all unpublished now, and got these things to kind of move around and showed they could either walk or roll or they could flip and do different interesting things. And when he came to our group meeting and showed this, everybody was very excited. And then I have another student who's called Eureka. And Eureka, about 1 month after Shin had shown this in our group meeting, came and presented his own group meeting, and he said, oh, Shin, I've done something even better with your kind of particle systems. Now I have them running up different staircases, and actually I've managed to make these particles be the fastest climbing speed that has ever been recorded for swarm microrobots. So this is kind of really interesting. And then what Eureka did was to take these kind of dual particle systems and make them slightly larger, and then they could do all sorts of exciting obstacles. And this is some work we're working on at the moment with Sharon Glotzer's group to kind of understand some of the mechanisms. Okay, so I'm going to talk a little bit about other types of drug delivery that are more kind of around the physical side of things, so thinking about how we might use mechanical interfaces, for example. And in particular, we're interested in getting things into cells using mechanical interfaces. So this is just an overview figure of some of the nanoscale technologies that people have used to extract content from cells. So for example, you can use glass nanopipettes or AFM tips, even carbon nanotubes, to extract small volumes from inside a living cell. It's really, really beautiful technology, but very difficult to scale, as you might imagine, and you can't use these things in the body. Then the technology at the bottom, which is nanostraw extraction from Stanford, is more scalable, but again, it's just more of an in vitro-based system. So work that we developed a few years ago now in collaboration with HMRI was actually the first nanoneedle-based system made from porous silicon needles that could interface with single cells. There's now many, many of these in the literature, but this was really one of the first systems that had been reported. And what we were interested in is thinking about how you could use these to interact in new and interesting ways with cells. And this is captured quite nicely in this review paper by Stuart Higgins, who's a postdoc of mine, who's now a professor at York. And what Stuart captured the summary of in this paper was how you could use these types of systems from our group, but also from other groups that followed with this work, and also separate groups that have been working on nanopillar systems, to measure things happening in cells, to deliver things with cells, to do interesting mechanotransduction of cells. And then finally, a big area we're working on is the bioelectronic and intracellular electrical stimulation and sensing. So if you're interested in this area, I would definitely refer you to this paper. And some of the things we're working on now are these sort of self-unfolding type needle systems. So this is what a cell on the bed of needles looks like. You'll see the nucleus appearing in blue here in a second. And what we find is the nucleus actually never gets penetrated by the cells, so it always moves out of the way. And you can see that here in this image. And in these particular needles designs that we have here, you can see the needle itself has this nanoscale porous structure. And that's pretty cool because it means you can measure things happening in the cell and you can also deliver things into the cell. And so Sohana, who was a PhD student in the group, did some mechanistic studies that actually came after our first big papers on these needles. But her mechanistic studies were very interesting because she actually did very high resolution electron microscopy imaging and also some biochemical measurements and showed that the membrane is still on the surface of these needles. It just becomes very convoluted, has a lot of clathrin pits and caveoli. You can see this in this image here, lots of invaginations. In the top right, what I was showing you is just the needles in green, and then in red is the sort of little invaginations that the membrane has formed on the surface of the needle. And this essentially showed us that we could get a lot more uptake of cargo than on a flat surface. And actually, I just saw my very good friend Alfredo Alexander Katz here that I didn't know was at this conference, but we together have published other papers too in ACS Nano and other journals and Nano Letters, where his lab has done molecular dynamics modeling to show that the effect of curvature essentially on the membrane increases the formation of these vesicle systems. And so we understand a lot about why these things are really efficient at getting cargo into cells. And this was the first paper where we demonstrated on the left both that we could use these to get cargo into cells in vitro, and then on the right, how we could actually use these needles to deliver, in this case, plasmid DNA into muscle, living muscle, and then you could get lots of new blood vessels to form 2 weeks later by kind of promoting the overexpression of a growth factor called vascular endothelial growth factor. And so this is really a platform technology. You can use it for many different things. We also know something very interesting about the way that cells mechanically sense these interfaces. The needles themselves are hard, right? But actually, because the cell doesn't attach to them in a normal way and doesn't form as many of what we call these focal adhesion points, the cell actually perceives it as a soft substrate, which is really counterintuitive. But it means that you have this very, very interesting interplay between the mechanotransduction signaling. So that, if you're interested in that area, these are some of the key papers there. Okay, so we can use it to also deliver larger things. So this work was first done by Chiro in our group. He's now a professor at King's. And what Chiro had shown is that we could take these needles, we could load them with quantum dots, and then if you interface cells on that, the quantum dots could get very efficiently into the cell, and you could use them then for different types of biosensing. And then Dan Hakim, who was a postdoc in the group, he's now a professor back in Chile, looked at how you could use these to deliver what are called these polyplex-based systems. So they encode for DNA as well, but within a polymer matrix, and that you could actually use these needles to deliver these particles by layering them in different layers on top of the needles, interfaced also with different types of polymers, as you can see on the right there. So you'd have a layer of polymer, and then your polyplex, another layer of polymer, and so on. So this is quite similar to the layer-by-layer work that people like Paula Hammond have been doing, but in this particular case, we apply it to the needle. And when you do that, what you find is that if you use these to deliver now to the heart, so this is real cardiac tissue data, that we show that these kind of systems are very, very efficient in enabling those polyplexes containing the DNA to get into the heart cells. And if you look at the bottom, you can see that if you deliver the polyplexes via the nanoneedles, you'll get many more in than if you just expect these polyplexes to get in there from a flat substrate. So this is a way of not only delivering locally from the needle, but also enhancing the way that other nanoparticles can get into cells and tissues as well. Okay, so I'm just going to wrap up the last bit of my talk by talking about disease detection. This is important for any disease you can think of, from cardiovascular disease to cancers to antimicrobial resistance and also viral infections. Maybe even monkeypox. Let's see how this goes. And we're using nanoparticle systems here. And these have been around a long time. If any of you have ever been to the British Museum in London, this is a really cool thing to go and see. This is called the Lycurgus cup. It's back from the 4th century AD. It's really old. And if you shine a light inside it, it looks red. You shine a light at it, it looks green. And that's because it has within the wall of it these gold nanoparticles that interact with light in interesting ways at the nanoscale. So what our group has been doing is working on different types of nanoparticle systems and really thinking about how we tailor the surface to do interesting types of biosensing. And we work a lot with Professor Jaworski, who always does a lot of molecular dynamics of how ligands are presented at the surface, and this enables us to iteratively optimize our designs. So this schematic here, which is from quite an old paper now from our group, shows the different types of ligands that you can put on the surface of a nanoparticle, and you can put many others as well, but it serves as a nice reminder of the kind of different measurement systems we've looked at over the years. So everything from detection of enzymes, proteases, phospholipases, kinases, acetyltransferases, to detection of proteins through antibody binding, detection of different nucleic acids and small molecules, aptamer binding, and so on. The surface of a nanoparticle is a very, very versatile place to be able to engineer for detection of different things that you might want to detect that might be related to a particular disease. And so I'll start off by showing you a nanoparticle system we designed that turns the urine blue if you have an early cancer. This is a gold nanoparticle-based system that detects the action of proteases, which are enzymes that can cleave through peptide bonds. And the sensor system looks like the top left there. So we start with a protein, and then that protein is called neutravidin, and that can bind 4 different biotin groups, biotin being a small molecule. And that biotin is attached to a peptide that has a small gold nanocluster on the end. And this is what the gold nanoclusters look like. And essentially, that system, when it's all assembled, is about 11 nanometers in diameter. And that is too big to come out through the kidney. So if you inject this into the body, it is not going to come out through the kidney till quite a long time after you inject it, about 24 hours afterwards. But what we have now is these small peptides that can be cleaved by protease related to the cancer. And when it does that, these small gold clusters are released, and they're now just 2 nanometers in diameter, and they're small enough to come out through the kidney. And they can collect in the urine, and then they can have a catalytic reaction when we add some different chemicals to that, and they will turn the urine green. And so you can see this is very, very, very clear color change. At the bottom left there, we have healthy mice, and in the top layer, we have mice that have an in vivo model of colorectal cancer. This is a model that Sangeeta Bhatia has in her group at MIT, and she helped us with these in vivo studies. And then you can see very clearly that this is 1 hour after we've delivered our particles to the mice. They are getting this very, very striking color change, but only if they have a cancer, because that's the only one that upregulates this protease, which is called matrix metalloprotease 9. Now, this is quite a long way from translation to human. It's just a preclinical model at the moment in this Nature Nano paper. And there's quite a lot of work that would need to do to translate this to human, but it's interesting as a sort of concept of how you can use responsive systems to help you with disease readout. The next work that I'm going to show you is actually, all of it has been tested in human. It's much further down the translational pathway. And what I want to really get across is that, particularly in the global south, there are a lot of diseases that disproportionately affect people, and where technology could be really doing a better job of making sure that the technologies that we develop can be accessible to as wide a population as possible. And so I'm going to be talking a lot about technologies we've been designing so that they can have the most possible, widest global access. And one of the ways that we do this is by combining our technologies with mobile phones. And so this is an area called mHealth. If you're interested in this, we obviously discuss this a lot in our Nature paper, but this is a technology area where you can really think about using the capability of mobile phones to help in mobile diagnostic setups. And this is actually a really, really good way of democratizing access to healthcare. There's now actually close to 8 billion global mobile phone subscriptions. It's gone up even since we published this paper. So if you can see in this paper here, we've got an increase in smartphone adoption across the world, but also in places like sub-Saharan Africa. And it's not just the number of smartphones that's increasing, it's their capabilities. So you all know this, of course. Your phones are able to take better and better pictures, and that's something that we can really use in the application towards democratizing access to diagnostics. So in this paper, what we did is we looked at people that could get to, this is data from Uganda, people that could get to a healthcare center or that could get to diagnostics that would be accessible to be read by mobile phones. And not surprisingly, if you can make diagnostics accessible by mobile phone as a readout rather than have to go into a healthcare center, you can access many, many more people. So this is a really important area. We set up this center actually over 10 years ago now, so before the COVID pandemic, but it's been really active during the COVID pandemic because it's a center focused around infectious diseases, thinking about how we can track population, symptoms within populations. This is all work led by UCL, not by my group, but by Ingemar Cox's group, in collaboration with Google and Twitter. And then my part of the center is around developing tests that we can read by mobile phone, and then we look at integration into online care pathways. Now, we've done lots and lots of work through the center. About half my group is working in this area, but I'm going to show you just a couple examples today of some of the work that I think is really transformative. And so this work here that was led by Colleen when she was a PhD student in the group was thinking about how we could develop actually the first test in the world that would be able to detect very early stage HIV without sophisticated instrumentation. Because the tests that existed at the time when we did this work were not sensitive enough to detect virus infection within a couple of days. You needed to wait several weeks till your body had produced antibodies to HIV. And what Colleen did was to develop these particles that have a gold core and a porous platinum shell. They're called nanozymes. They act a little bit like biological enzymes, like horseradish peroxidase, but they're much more robust to extremes of temperature. That's going to be really important if we're taking them, for example, out into Africa. And they produce this really strong color change by oxidizing TMB substrate. And so you can use them the same as you would use a gold nanoparticle in a COVID test, for example, where you see that red line, but you're producing a much stronger color change. And that means you can detect much lower concentrations of your target. So we can take these particles, we decorate them with antibodies that can recognize this protein called P24, which is on the surface of the viral capsid for HIV. And then we have another small nanobody functionalized with biotin, and that can also bind to the target. You can then run these up a test strip. This is like a lateral flow test strip, and the biotin binds very quickly to streptavidin at the test line, and then you can essentially oxidize your substrate into this colored product, and you end up with 100-fold amplification. So this system actually ends up being 100 times more sensitive than the traditional gold lateral flow test. And you can see our system there in the red that's been amplified was the first system ever to get into the early stage of HIV detection. And this was published a few years ago, but is now in this product called Quixzyme, which is in early commercial manufacture, and it's being applied also for other applications in cardiac field and also in the cancer field, as well as some projects that we have in cholera detection, so with Bangladesh, and then a tuberculosis and malaria programs as well. Final piece around the nanozyme technology is we've also combined it with this area called CRISPR diagnostics and formed this assay called CRISPRzyme. And this is essentially a technology that can detect different types of long non-coding nucleic acids. And this is data from where we looked at this with patients that were presenting to the emergency room and had potential heart attacks. And what we were able to do with this technology is actually determine whether the patients were having a real heart attack or not, but very quickly without needing sophisticated instrumentation. And again, that is just not possible currently with other technologies. And the exciting thing about it here is that both equipment, we don't need equipment for these kind of technologies, so you save a lot of costs there, and consumables are very cheap as well. And this is all now within a company that has a very strong drive to provide social impact and these kind of diagnostics within low and middle income settings at cost. We work a lot with our collaborators in Africa. In particular, a lot of the work we've done in HIV has been in the Africa Health Research Institute, and also have taken some of this work out to Uganda. In this case, during the last Ebola Gulu outbreak, we were able to do this geotag mapping of 3 different strains of the Ebola virus. And actually this paper, I put it here because it was led by my PhD student, Paulina Brangle, during her PhD, and it was one of the first examples actually of this geotag serological surveillance that we're all probably now very familiar with after the COVID pandemic, but this was before that. And what Paulina ended up doing after her PhD is she ended up going to work at the WHO, and she became the person in charge of doing serological surveillance for the COVID pandemic. So this actually was a PhD that ended up being very useful later in real life as well. I just finished with this video from iSense. So this is showing some of the work that's been done also at UCL, creating these carrier systems so that we can have HIV tests within there with robust markers, so you can get really accurate pictures and readouts. And this is all then put into these data dashboards that were also developed within iSense to get quicker integration of patients into care in KwaZulu-Natal in South Africa. So I'm going to stop there because I do want to leave a couple of minutes at the end. But thank you so very much to all our funders, all the people, all the audience here, and the people who've invited me as well. Thank you.
Thank you very much. Thank you, Molly. That was certainly a very nice overview about the different methods to functionalize materials and for diseases detection and treatment. So now the talk is open for questions. We have plenty of time, and may I remember you so that after this talk we have the coffee break. So please, could you please remain in the room? Because okay, please here.
Thank you, Professor Molly. Very nice presentation. I have a couple of questions. First one with the Spartan. For example, you're using benchtop Raman for samples, and now you're telling you're even having some kind of collaboration with pharmaceutical companies. Question is, for example, when we have samples with nanoparticles or those kind of structures with biological analytes, so we have to take care of the laser excitation source and the time we are doing for the measurement, and you have one algorithm going on the backside. The question is how much time it takes to do the calculations and to do one measurement, because for industrial scaling, I think that's very important, right?
Yeah, so I mean, that's a great question. And I'd say a lot of the work we've been focusing on after inventing the system has actually been on the software that can automate that and make that very user-friendly. And so that also includes a lot of calibration steps within that, includes a lot of analytical platforms, as well as actually reagents that we can supply alongside it that can help us with understanding sizing, charge, and all these other things that pharma would also be interested to understand alongside. So our aim really is to automate as much of that as possible so that things like the background and all of that you know are of course an issue in Raman are automatically removed without the user having to do those steps.
Thank you. The last question is, in the case of microneedles, for example, I remember that paper you have presented, the porous silicon structures for generation. The question is, because porous silicon, it tends to oxidize very fast. So how did you take care of that parameter, and also the break of the nanowires or these kind of nanotubes during the interaction?
Yeah.
Can you comment on that?
Actually, so the Nature Materials paper in the supplementary information section has a section around the kind of mechanical forces needed to break the needles. And the forces actually that are needed for that are far, far higher than what you would ever encounter in tissue interfacing. So that's not an issue, and all those calculations have been done. And then the material we make them from, actually we've now made them from many, many different types of materials, including polymer-based systems and different coatings and everything else. So I'd say that's very, very versatile. So you can just adapt it for different platforms you might be interested in. Yeah.
Thank you very much. Please, could you please share the microphone here? Thanks. Professor
A brilliant talk, Amale. My only critique is I didn't see any Ukrainian students in your group.
I think you need to help me get some, Yuri. Yeah.
Thank you. My question is about this field, pretty large actually, microparticles here. There seems to be like tens of microns inside, if not 100 of microns here. What kind of delivery are you considering? What are the basic applications for those?
Yeah, I mean, we have actually systems ranging from, 50 nanometers upwards, right? So we have very small particle systems, and we have some that are larger than that. So it really, really depends on the different application. I mean, we have projects that are more involved around systems that need to circulate a long time and then, get through kind of leaky capillaries and things like that. So those are obviously have to be typically 100 nanometers and less, and then larger systems that you might apply for different areas. So some of those porous capsules I showed you actually have been funded by the Gates Foundation with the idea that we might look at pulsatile vaccine systems. And so the idea is we inject them, they're not circulating through the body or anything, but they can then release cargo on demand at different times. So that's the application of that particular work. Yep.
Thank you.
Thank you, Yuri. Please, Cecilia.
Thank you for your talk. I'm wondering, for example, in this kind of application, you need to be very specific and for the response, and also, for example, to use like drug delivery and so on, you need to have always the same response, no?
Yeah.
So how much you can have variations of the particles, and so Which are the errors in this kind of
Yeah, I mean, I love that
system?
question because actually the whole reason for us developing the SPARTA technology was so that people could better understand their systems, so that they could understand heterogeneity, and then they could have more controllable responses. Because I think it's a real problem within the field as a whole that, I mean, not our printed nanoparticle systems, because those are very precise and very controllable, but other systems that you make in bulk in solution, for example, you know, you're looking often at a bulk response, and it might be different with, and also you have variability within biological systems as well. So, you know, as you mentioned, the sort of robustness of response is something that often has a lot of noise. And so what we wanted to do was essentially develop technology so people could understand where the noise was coming from. Is it from the formulation itself? Is it that sometimes you're loading drug differently? Is it sometimes your particles are not degrading at the same rate? Is it that they're absorbing proteins differently? All of these kind of questions that you cannot really adequately answer from bulk measurement techniques, you can now answer once you start to study at the single particle level. So I really hope this SPARTA technology will be enabling for that whole field in terms of much better mechanistic understanding of robustness of the system, essentially.
Okay, thank you. I've got myself a question related to the needles bed. Do you guys fabricate the sensors, I mean the needles, because of there is a bunch of possibilities you showed, not only the needles bed, but also these AFM hollowed needles where you can either extract or inject different mechanisms. Do you have facilities to fabricate them?
Yeah, absolutely. So the figure from the Science article where we showed the hollow AFM and everything, as I mentioned during the talk, these are not technologies from our lab. That's kind of capturing an overview of the field. What we make in our lab are different nanoneedle and microneedle platforms, and we have a range of different technologies to do that. So the original nanoneedles from the Nature Materials paper were made using conventional microfabrication, but with a special metal-assisted chemical etch to get that kind of nanoscale porosity patterning. But we now make them from lots of other materials, including polymer materials and also different microneedle systems using different sort of templating and molding approaches, so as well as printing-based approaches.
And a very short last question. I was wondering about the size of the particles, because in most of the slides, so the scale bar was not, there was no number. Is there a reason?
Well, I mean, similar to Yuri's question, really, they're different in different papers. Yeah, so some of the sort of degradable particle systems are very small, 50 nanometers upwards. Others are more in the micron size. Yeah.
Thank you very much. So we got time for a final question. So please, Mildred.
Thank you for the very nice talk. The Raman machine that you presented is an optical trap, and there is something else to enhance the Raman signals of the biological things, because I think it's very similar, all the organic stuff in the Raman. How can you distinguish the difference?
Okay, that's a great question. So it depends on the application. So sometimes, in fact, very often, the native signal is fine, and we've published, for example, differences in cancer-derived exosomes as well as lipid nanoparticles loaded with drugs and all sorts of other things without enhancement. But we also have a whole area of research around surface-enhanced Raman reporters that can bind, for example, to particular receptors on a self-surface-derived exosome, or they can bind, to particular targets on a nanoparticle, or also be used to label sort of cargo. So a lot of it can be done label-free, but there is also the option to enhance particular components if you want as well. And we have technology also patented technology in that area. It's an area we call Raman bodies, so you get essentially targeted enhancement of a particular signal as well. Thanks for asking that, because I didn't mention that in the talk.
Yeah. Okay, I'm pretty sure so we will have more time, more questions and time after the finishing the talk in the coffee break. And so again, could you please join me to thank Molly for the very nice talk.
Thank you. Thank you.
And Jesus.
Thank you very much, Molly. Please, on behalf
you.
of the Mexican Material Society, receive this craft. It's made in this region. It's, as you can see, there is small balls there. We call it Shakira balls. They are 1 millimeter diameter, and they are placed one by one. So they take a little bit of time to do this.
It looks like self-assembled nanoparticles to me, but yes.
Yeah, actually, it looks like,
It's
right?
a very fitting gift. Thank you.
But it's unique in the world. You have
the only one of this.
The only one of these.
Yeah.
Thank you very much.
Thank you so much. Thank you.